Next, we'll have Sandeep from Chen's lab will talk about TissueMosaic, which uses self-supervised representation learning to enable cross-sample differential analysis of spatial transcriptomics dataset. Welcome.
Hi, I'm Sandeep. I'm a graduate student advised by Chen and Kambhampati, and I'll be talking to you today about our recent method, TissueMosaic, which stands for motif-based spatial inference across conditions. And so this is a model that can learn representations of spatial transcriptomics data via self-supervision, and then these representations enable cross-sample differential analysis of ST data. So just to give an overview of what ST data is, this is a powerful experimental platform that can measure gene expression in native tissue context. And what this allows us to do is start to study how cell state is sort of modulated by a cell's local environment. And so the way our data is structured is for a given sample, we'll have a list of spots, say 1 to n, and for each spot, we'll have its XY coordinate and the measured gene expression profile at that spot. And so one sort of common downstream task of interest with gene expression data is differential gene expression analysis. Let's say you have a set of samples from healthy and diseased tissue. You can sequence the mRNA, and then you can identify genes that are more highly or more lowly expressed in your diseased samples. And these are genes that are presumably implicated in the underlying disease process. And so we sort of set out to develop a similar method to enable differential analysis of our spatial data. And so more concretely, if you have a group of samples from control, another group of samples from case, how can we start to understand how tissue structure changes across these two conditions? And so one issue with sort of answering this question is that we need to define what the basic unit of tissue structure is, the way that genes are a unit of comparison for bulk RNA-seq data. And so if we could define a similar unit for our spatial data, we can then start to compare samples across multiple conditions. And so in our work, we call these units tissue motifs, and our goal is to discover these motifs in an unbiased way. And we heard some really cool talks yesterday about the importance of learning unbiased niche representations and how these can enable differential analysis across control and case samples. And so in a similar vein, what we want to enable is a common representational space that can unify our entire spatial data set, such that if you extracted units or say patches from these samples, you can then embed them in a common latent space. And you can start using that latent space to compare and contrast tissue structure across samples and conditions. And one thing I want to sort of highlight is the spatial heterogeneity that's inherent to tissues. So you can imagine in this latent space, we might have, so in this latent space, like sort of a motif, as we defined earlier, sort of like a local organizational pattern that appears. And so you can imagine some motifs might be shared across control and case samples, whereas other motifs are sort of enriched for control versus case samples. And so in our differential analysis procedure, what we might want to do is sort of focus on the motifs that are overrepresented in our case samples. So this then introduces the next question of how do we learn these representations without any underlying labels for tissue structure. And so to do that, we turn to self-supervised learning, which is a sort of framework where you can define a proxy supervised task to train your model. And in solving that task, the model will learn meaningful features that can then generalize to other downstream tasks. So one common framework in computer vision is contrastive learning. And just to build some intuition, the idea here is that let's say you have an image data set without any class labels, but you still want to train a classifier. What you can do is you can take each image in your data set and then perform 2 random augmentations of that image. And then you can instruct your model to produce representations that are similar to the 2 input augmentations and then dissimilar to the other images in your data set. And so in solving this task, the model will sort of learn what it means for a dog to look like a dog and how a dog might differ from, say, a chair. So sort of inspired by the success of these class of approaches on natural images, we decided to take a similar approach for our spatial data and model them as cell type images. And so what we do is for each spot, we take its XY coordinate, and from the gene expression, we can infer the cell type or cell type proportions at that spot, and then we can rasterize that information into a multidimensional image where each channel of this image corresponds to a different cell type. And so on the right, we have an example projection of what that image would look like for some mouse testis tissue, and we can sort of see here the emergence of tissue structure in the form of these tubules. So once we have these cell type images, we can then apply random augmentations to them. And so some of the augmentations that we choose are ones commonly applied in computer vision, like resize cropping, flipping, rotations. But we also design some augmentations that are tailored to our spatial transcriptomics data. So one example of this is channel or cell type dropout. And this sort of forces the model to pay attention to information that's present in all of the cell types in your data, regardless of how rare or abundant they might be. Another augmentation, this straight cut augmentation, sort of desensitizes the model to any edge effects you might introduce by sort of sampling a square patch at the edge of a circular tissue sample. And so we sort of have a set of 10 or 11 different augmentations, and some other augmentations can desensitize the model to different technical artifacts in your data, which helps us overcome some of the batch effects when we want to integrate multiple samples together. And in each training pass, we can extract a patch from this image where the size of this patch is a user hyperparameter and sort of reflects your expected length scale of your tissue. And we can randomly select a set of augmentations and then compose them together to produce two augmented views of your input patch. And so then the way we train our model is like, given these two input augmentations, we can pass them through two different branches of our network and then use this cross-entropy loss that sort of encourages the model to produce representations that are similar for the two input augmentations. And so this self-supervised learning framework is called Dino and has sort of shown great success on natural images. And once we've trained this model, we can take this trained ResNet, which is our feature extractor, and we can tile our sample, our data set, with a large number of patches, pass each patch through our trained model, and then produce a set of representations that then encapsulate the local tissue structure present in your data set. And so then we can start to discover tissue motifs that are present in our data. And so this approach that we call TissueMosaic enables several downstream tasks, including motif query, gene expression regression, and motif enrichment. And I'll briefly go through these three tasks on two different spatial data sets. One is of the mouse testis and has 3 wild type and 3 diabetic samples. And the other is of the mouse, is an atlas of aging in the mouse thymus. So the first task I'll talk about is motif query. And the idea here is that given an input patch, we want to find other regions in our samples that share the same motif as this query patch. So in the testis, there are sort of 2 main motifs. We have these RS tubules in orange and these SPC-rich tubules in red. And so we can see that if we query the sample with an RS-rich tubule, we're able to retrieve other RS-rich tubules, where here we're plotting the similarity of the retrieval representation to the query representation as an alpha transparency mask. So the more opaque the retrieval is, the more similar it is to the query. And so what's sort of notable here is that despite using square patches and no other segmentation input, the model is starting to able to learn the circular boundaries of these tubules. We can do something similar with the SPC-rich tubules, and so we see here with just a single example of each query motif, we're able to separate our sample into the 2 main motifs present in the data. And so another task that's very related to motif query is spatial clustering. We can discretely group the learned latent representations to get spatial clusters. And so in the mouse thymus data, the 2 main anatomical regions are the cortex in blue and the medulla in orange. And we can see that when you cluster the tissue mosaic representations, we get really good concordance with these ground truth manual annotations of the same samples. And then we can quantify this performance by a normalized mutual information metric. And we can see that we can outperform this baseline that's just based on the cell type compositions of the patches, like treating each patch as a bag of cells. And we can also outperform several other existing representation learning clustering methods. So sort of convinced we were able to learn some meaningful representations of tissue structure, we then wanted to connect these representations to the underlying gene expression through a downstream regression task. And so here I have this regression model for those who are interested, but I won't go into detail. But just to build some intuition, what we're trying to do here is we want to identify genes that sort of co-vary with their tissue motif representations. And essentially identify genes that have a high mutual information with tissue structure, which we sort of formalize with this motif information score. So if we run this framework on our testis data, we can get this distribution of information scores across genes. And we can see that a gene with a low information score, like FABP9, we're also doing this conditioned on cell type, so within a specific cell type of interest. So conditioned on ES cells, we can see a gene like FABP9 has a spatially random distribution of expression, whereas a gene like TNP1 that's at the tail end of this distribution has a much more structured distribution of expression. And so what's cool here is that even though you can't easily parameterize this spatial pattern, the model is still able to pick it up as having high mutual information with tissue structure. And so we finally wanted to return to our sort of motivating task, which was the differential analysis of spatial data. And this is a task that we call motif enrichment. And the idea here is that given a training set of motif representations and their condition labels, we can train a classifier to predict condition label from motif representation. And then on a new held out sample, we can identify different motifs that are similar to the motifs in our training set that are enriched for different conditions in our training set. And then we can start to do differential expression analysis at this motif level rather than the sample level. So when we run this framework on our thymus data, one hallmark of a gene in the thymus are the presence of these antigen-presenting cell aggregates. We can see the model actually highlights these aggregated APCs as having high biological age. And so what this enables us to do is to then run differential expression specifically between the aged APCs and the non-aged APCs. And one gene that this analysis highlights is CD74, which has been previously implicated in malignancies of these cells. We can also go beyond just magnitude of expression, and we can also start to identify genes that are spatially dysregulated across samples from different conditions. And so we demonstrate this in the testis, where one hallmark of disease is the presence of these disorganized tubules, where you kind of see that the periphery cells that are orange or red sort of get mixed with the blue cells on the interior. And so we can quantify this disorganization with this ES purity metric. We can see the model is able to highlight the impure ES cells as having a high diabetic scores, which is we think is a fairly subtle phenotype of disorganization in this data. And what's kind of cool is that we can then use our regression framework to define this differential tissue motif information score. And so this score is essentially sort of a metric of spatial dysregulation across conditions. And so in blue, this is a distribution across genes. And in blue, we have, when we run this regression framework, just using sample label. And then in red, we have this distribution when we run it using the enriched motifs, so kind of focusing in on the regions that are enriched in disease versus healthy samples. And we can see that a gene like SMCP that's known to be dysregulated in the testis during diabetes has a small negative differential score, but by performing this motif enrichment procedure, we can detect a much greater effect size for this gene. And we also developed a hypothesis testing framework that can allow you to assign statistical significance to these scores. And we think that this sort of result is kind of powerful in sort of starting to identify genes and gene programs that are sort of spatially dysregulated within diseased samples. And so in conclusion, here we have TissueMosaic, a method that learns representations of tissue structure and connects these representations back to the underlying gene expression. And then by doing this, we're able to enable cross-sample differential analysis so we can identify motifs that vary across, say, healthy versus disease, and then furthermore detect genes that covary with these motifs. And so by doing this, we can then see how changes in tissue organization, say due to disease, start to impact transcriptional programs and start to sort of identify the functional organization of these tissues. And so with that, I'd like to thank my mentors, Faye and Murtash. If you want to learn more about our work, we have a preprint out. We demonstrate this in a variety of different technologies and tissues to show the model readily generalizes to different data sets. I'd also like to thank my co-authors, particularly Luca, who made a lot of contributions to the code base. And yeah, I'd be happy to take any questions.
Great talk. Thanks, Sandeep. We have room for one question before our coffee break.
So when you do this dino-like thing and did you come from the same sample, or were you the augmented versions? Are there other similarities so that you could take things from different regions of different images and still have like maybe an external label that would let you do that cross entropy?
Right. So you're saying instead of taking the same, like you can contrast against patches from different images.
Yeah, like in the past we've done things where are you the same fold? Are you the same fold and function? And of course you then end up doing a ton of work to figure out which of those relationships are reliable enough.
Yeah.
But have you thought about that so that it's not just are you two augmented versions of the same patch, but you're actually two other things that are substantively similar across some other annotation?
Yeah. So we thought about that a little bit in the context of a different loss, like Sinclair. So here with this dino loss, you kind of we thought about using the frame cross Sinclair, where you have like negative samples you contrast against, and then maybe try to use that to like overcome batch effects and things like that by contrasting against maybe like patches from like you can maybe like specifically only have patches from the same sample that you contrast against, and maybe then try to overcome sample specific effects that way. But actually what we found was that just this we didn't need to worry about doing any of that. Just this like straightforward implementation with taking the same patch actually worked pretty well at learning some integrated embedding space. And we think one reason for that is that we're just modeling cell types, and so those cell types are shared across all of the data. And so that's kind of how we get at that.
Okay, so thank you to all the speakers of this morning's session.